Okay, so we find ourselves in the beginning of Chapter 2. And in Chapter 2, we have some really interesting discussions about a broad topic called pharmacodynamics. And pharmacodynamics is a word that refers to what a drug does inside the body. Right? What the drug does to the body. That's pharmacodynamics. As opposed to pharmacokinetics, which is what the body does to the drug. So you kind of have like this constant interaction between what the drug's trying to achieve in the body and then what the body's doing to the drug. So you're administering a drug to a patient, and their body is already starting to eliminate the drug, right? And so there'll come a point where a patient will have to take another dose of the drug because their body would otherwise eliminate the drug, right? So there's like a kind of a pull and push mechanism of sorts when you look at these terms. And so the way your book is organized is first it talks to you about pharmacodynamic principles, and we actually spend about 3 lectures in that chapter. And then it talks to us about pharmacokinetic principles in general terms. So things like drug absorption, drug distribution, drug metabolism, drug elimination. And we pretty much talk about that in 2 lectures. And then we have a whole special lecture dedicated to metabolism, to the M part of ADME. Right? We talk about how metabolism works. And really it becomes relevant to us because we have patients who are taking drugs and whose metabolism could be different due to genetics. So you may have heard of this field called pharmacogenetics, where alterations in particular drug metabolizing enzymes can really alter drug responses in a patient. Right? So if you give a drug to somebody and they're a poor metabolizer of that drug, you can get into toxicity issues at some point. And sometimes the toxicity might be self-limiting, like they pass out and faint and end up in the doctor's office or the hospital. Sometimes the toxicity is more insidious and hurt them long term. So we spend a whole chapter in our book, as you kind of look forward, chapter 5, looking at this field of pharmacogenetics. So that's kind of where we're going with the principles. And then that will kind of bracket the principles, which then stay with us as we move forward into any drug system. Right? So once we get through all that stuff, it's midterm time. And there's really not that much time until midterm time. So we're going to get through all that stuff, and then after the midterm, we're going to apply those principles to the drugs that affect the autonomic nervous system. So that's kind of the structure that we're following. And so today, I want to talk about this topic of, this first topic in Chapter 2 of receptors. And in the present day, most of us have heard the word receptor. It's kind of become part of the common language of drugs and how drugs work. But it wasn't always known that drugs work through receptors. Right? Receptor theory is just about 100 years old. This is about now. And receptor theory gives us the link between drug and cellular or tissue response. So realize that this is a cell that I'm drawing here in this little circle. The receptor, where part of it's on the outside, part of it goes through the membrane, and part of it's on the inside. Oftentimes the response is on the inside. Not always, but often. And so how is it that drug binding out here leads to response in there? What's the connection, right? And so for many years we didn't really know the connection, but now we know that there are receptors that are the first response in drug action. And so when a drug binds to a receptor, something happens. Right? So the receptor can be defined down here below as the component of the cell or organism that interacts with the drug and that initiates a chain of biochemical events that leads to the drug's effect. Right, so the drug binds to the receptor, and then we have a chain of biochemical events. And so the chain is what's going to really be different from one receptor to another. Right, so the chain can be very lengthy and have multiple steps. The chain can be very short and maybe have one or two links on the chain. Or in some cases, there's just a single link. The receptor is the chain. So the drug binds, there's a change, and the response happens immediately. Right, so the question is, when you go from drug binding to a receptor to response, right, when you go from drug binding to a receptor to a response, how long is the chain? Is it a single link? It's got a few links? Or does it have a lot of links? Right, so that's what we talk about in our next lecture, the links of the chain and how we can go from drug binding to response. But for the moment, the receptor is that which binds the drug and initiates this chain of events. So the chain of events might be small, the chain of events might be complex. And we'll study this in detail as we move forward. Okay, so it's the component of the cell or the organism that interacts with the drug. And what's important about the receptor is its nature. So the majority of drug receptors are what we would call macromolecules. Macro meaning big, molecule meaning molecule, big molecule. Right? So most of these receptors are macromolecules. And so, can you name a big molecule in cells? Humongous, potentially large and humongous molecule. Is glucose a big molecule compared to something like insulin or a big, super large peptide receptor of some sort? Right, so peptides and proteins are big molecules. What else is big? They contain nucleic acids. DNA. Very good, right? So DNA can act as a receptor. In some cases, RNA can act as a receptor. And so, when we talk about these macromolecules, we're talking about really big molecules. And changing them in some way will initiate this chain of events. So, we know that the receptor determines the quantitative relationship between the dose of a compound or drug and its pharmacologic effect. Okay, there is a quantitative relationship between dose and effect. And so what does that look like? So remember last class we talked about pharmacology, experimental science. Right? It's an experimental science that has for its purpose the study of changes brought about in biological systems by chemically active substances, whether for therapeutic purposes or not. And so part of that definition is experimental science. And so for over 100 years we've had pharmacologists study compounds and their effects in cells and tissues, right? In vitro science. And basically what they see time and time again is a quantitative relationship between dose and effect, between concentration and response. Now, I don't remember if last class I talked to you about the difference between concentration and dose. Right, so just to review, concentration is going to be an amount of a drug in a certain volume of fluid. Right, so if I have cells growing on a tissue culture plate and I have fluid above them, bathing them, hydrating them of the right consistency, right? Sugar and spice and everything that has salt, has glucose, has osmotic properties. All those things that balance with the cytoplasm of those cells will grow. So the concentration concentration of a drug that we might add might be 1 milligram to a volume of 1 milliliter. Or it might be 5 milligrams to a volume of 1 milliliter. It might be 10 milligrams. So concentration is going to be milligrams per milliliter, or milligrams per deciliter, or micrograms per deciliter, but some kind of amount or a volume. So this is what we do in vitro. We add a very exact amount of compound to the fluid. We know how much compound is in the fluid. In an animal model, we usually talk about dose. Because in the animal, we're administering a certain amount of drug, but what's the denominator? Right? What's the denominator in the animal? Of course, you have blood volume. Some of the drug obviously gets absorbed into the blood. The drug actually also leaves the blood and enter parenchymal cells, right? So the blood is like the highway, and it's like when you drive home from school, you drive on the highway, and then you drive on a side road, which would be like a capillary bed, and then you drive to your front of your house, and then you actually get out of your car, right? You enter the house. It's like a drug gets out of the blood and enters the cell, right? So yeah, there's blood in tissues, but blood is a conduit for drug permeation, right? For drug movement. But drugs won't always be in the blood after you administer them. Blood's carrying them around, and they're going to use out of blood in the tissue. Do something in the tissue, get metabolized, end up coming back into the blood, getting carried to the kidney, and excreted in the urine, or ending up in the bile and being excreted in the feces. So once you give something to an animal, you don't exactly know where they went. When you put 10 milligrams in a little solution in a culture dish of 10 milliliters, you exactly know where the drug is. It didn't go anywhere. Some of it went into the cell. It has volume. There. And when you give a dose to an animal, this is going to be my very poor picture of a needle. And maybe you've given 10 milligrams to the animal. Where did it go? You're going to measure some of the urine, you're going to measure some of the blood, some of it's going into fat, some of it's going into muscle, whatever, right? So much harder to know where it goes. So you can't really say milligrams per milliliter unless you're specifically talking about the blood. You never realize what that blood. So when it comes to animals, we use this term doses. So when you go in your pharmacies on rotation or working there in the summer, you usually give like a dose of a drug to somebody, right? Like you give them the 10 milligram tablet. You give them a 100 milligram tablet. You give them 1,000 milligram tablet. A few micrograms bound into the tablet. You're not thinking in that moment, 10 milligrams per milliliter concentration and dose. So do you feel comfortable? The difference between a dose, which is an amount, and a concentration, which is an amount and a volume, that you know the volume of. Okay? So that's why in pharmacology sometimes we'll say dose response curve. The dose response curve applies to then what? Response in an animal. If we say concentration effect curve, we're looking at an in vitro experiment on cell and tissue. So our book in chapter 2, I just started to teach through it, and I definitely urge you to look at it, even if you don't always go on the Access Medicine website and read it. Right? You'll see lots of graphs in chapter 2. And so the graphs, first thing you should ask yourself, well, what is this? Is this dose? Is this concentration? That'll tell you if I'm in an animal model of some sort, a mouse, pig, human, or I'm in vitro, cells and tissue. And so now going back to this situation, this is an experimental science, right, where we're going to look at the concentration of drug. What does brackets mean in chemistry world? Concentration, right? So concentration of drug versus effect. Whatever the effect is I'm measuring. Well, maybe I'm measuring inhibited rate of contraction of heart cells in culture. Maybe I'm measuring relaxation of some muscle in culture. Right? But whatever I'm measuring, what pharmacologists have known for some time is that if I give a little bit of drug, I have a little bit of effect. If I give a little bit more drug, I have a little bit more of an effect. And if I keep giving a little bit more drug, I get a little bit more of an effect. Until eventually, I get to a point where even though I'm increasing the drug, I'm not increasing the effect. Right? So the curve looks like a hyperbola. And so I'll get to a point where I can have this amount of drug, and the effect is here. And I might double, triple, quadruple, 10 times, 100 times the amount of drug, but yet my effect is no longer going up. Right? It has plateaued. Right? We call it the plateau effect. We can say we maxed out. Right? It's kind of like when you're in the gym and you're doing a bench press. You get to the point where you're maxed out. That's it. You can't do it. Right? So that's kind of how drugs work with targets. There'll come a point where the effect is maxed. And even if you give more drug, you won't get more effect, and you'll most likely start to get toxicity, which will be a topic of 2 lectures from now. So if we were to look at this, we know that this is a quantitative relationship. There's a relationship between the concentration and the effect. And we now know that this relationship is driven by receptors. Right? These macromolecules that bind to the drug. And so this relationship is going to be true of drug versus effect. And it also happens to be true if we were to just change one of the endpoints we're looking at. And rather than looking at effect on the y-axis, we look at receptor binding. Right? If we look at receptor binding on the y-axis versus drug concentration, we usually see the same kind of effect, same kind of effect. A little bit of drug, a little bit of receptor binding. A little bit more drug, so more of those receptors in the tissue are bound. A little bit more drug, I start to bind up more and more. But eventually I'll get to a concentration where I've bound up all the receptors that I can. Right? So if this is a tissue, it doesn't really look like a tissue, but if it's a tissue with receptors on it, right, there'll come a point where I bind up all of the receptors. Let's call that the 1x concentration, whatever that is. And now let's say, rather than the 1x concentration, and I'm looking at binding, right, so here 100% of the receptors are bound, right? So now, instead of the 1x concentration, I give 10x concentration. What percentage of receptors are bound? All of them. What if I give 100x concentration? What percentage of the receptors are bound? All of them. What if I give 1000x concentration? All of them, right? So I get to a point where I can't bind more than are there, right? And so if we were to look at that curve, we can see a little bit of drug, a little bit of binding, and eventually we saturate. Sometimes it's called saturation. Right? We saturate all of the receptors. And so on a drug receptor binding curve, we call this portion of the graph the plateau. We call it the Bmax, the maximum binding. Okay? On a concentration effect curve, we call the plateau the Emax, or the maximum effect. And what I tried to draw here, and it's not exactly perfect, but what I tried to draw here is that usually maximum binding corresponds to maximum effect. It's a quantitative relationship. So in your notes, I would say this usually is what happens. Right? So the concentration where I bind all my receptors is the concentration that gives me maximum effect. Usually what happens. And then put a comma and say, except when I have spare receptors. The spare receptors are an exception to this rule, and it'll be something I talk about later. It's an exception to the rule. Okay. So most cells and most tissues work like I've been spending the past 10 minutes talking about. And then there's an exception to the rule called spare receptors. And I'll just give you a precursor to that. In spare receptors, you can have a maximum effect without having all the receptors bound. You can have a maximum effect at concentration where you don't have maximum reception. And that will come later. So receptors are going to determine this quantitative relationship, point 1. Point 2, they're going to be responsible for selectivity of drug action. So as you probably learned in med chem, right, receptors are going to have a region that binds to a drug, called the drug binding domain. Right, it's going to be the part of the receptor that binds to the drug. The drug binding domain. And the drug binding domain is going to have physical limitations. Right? It's going to have a certain structure. Right? So it's like when you start one day down the road looking to buy a house or an apartment somewhere. You're going to go looking around and you're going to see different three-dimensional structures. Right? You're going to see colonial houses. You're going to see capes. You're going to see condos. You're going to see 3-bedroom apartments, 2-bedroom apartments. They're going to be fixed in how they look. There's a certain look to them. So unless you demolish them and build something that you want, you're kind of stuck with structure in front of you. Right? You paint the walls, you can put some wallpaper in the bathroom. The structure is the structure. And so with the drug binding domain of a receptor, it's going to have a certain structure. And so only certain drugs can fit into it, like a lock fitting into a key. Right? So things like size of the binding site, shape, electrical charge. Right? If I have a binding site that has a lot of negatively charged amino acids in it, right, I can't really have a drug that has a lot of negatively charged ions facing that part. That would be a bad fit. But if I had a binding site that had a lot of negatively charged amino acids in it, and my drug has a lot of positively charged amino acids in it, that's a good beginning partnership. Right? So something about size, shape, and charge is all going to have to come together appropriately for the receptor to bind to the drug. Why one receptor binds one drug or a few drugs and not thousands. It's also why, as you may have learned in medicinal, why some drugs, when they're synthesized in chiral centers, it ends up causing formation of an enantiomer, so you have like an L or a D enantiomer. It's also why sometimes only one of the enantiomers binds to the receptor, because the other is not superimposable. It doesn't fit the same way. Right? So some drugs are synthesized as a racemic mixture. Right? But only one of the racemates actually binds to the receptor, the other is the inactive. In some cases, change due to the L or D is not severe enough to affect receptor binding. So it depends. But drugs that have chiral centers may have more active enantiomers than others. And I'm sure you've talked about some examples of this in med chem, but just by some chance you haven't, there will be examples of drugs like this, and you totally agree. One of them is warfarin, right? It exists in multiple enantiomers, such as diastereomers, and some of them more active than the other. But drugs usually just dispense in the so what that means is it's still okay for the right? So you may have learned in med chem that separating enantiomers is a challenge. Right? I don't know if you remember Louis Pasteur, he worked in the wine industry in France, and he's the one who discovered L and D, right? Levothyroxine dextrose. What he did is he sat in the microscope and looked at sugar grains. They actually saw that they looked different than each other. They separated them out with group of ethers, two files. That's really thinking of one kind of different. They separated them out and he dissolved one group in water, dissolved the other group in water. They were both hydrophilic. Then he shined light through the water and found that one of the samples rotated light in one direction, while the other samples rotated light in another direction. This idea of isomers. Since then, there's been a lot of work on that. this because, why I'm mentioning this is because it's labor intensive to separate out an antibodies. It's not two look very similar, so it's hard to pull them apart. Not impossible, but it's hard. So if from a manufacturing point of view, the receiving mixture works, why would I add cost to my manufacturing process to separate them out? Unless, of course, the patent is expiring on the mixture, and then I can extend my patent life by purifying more active antibody and saying it's a more potent drug, going to the FDA, and that's years. Okay. So anyway, I think you understand that the drug receptor is going to selectively bind to the drug, and that these are the sites of binding of the pharmacologic agents. Now, today, in 2015, we have a clear view that most of these macromolecules that act as receptors fall into the protein or peptide category. That wasn't always the case. So, if you go back 50 years, right, the technology was different. And so 50 years ago, the way new drugs were discovered by taking old drugs, certain drug, we know how it works, by modifying its structure. Right? So let's stick a cyclohexyl group on one end, or let's put some hydrogen-containing group over here, or let's put a nice propofol here, a cyclohexyl ring. Let's do this structure activity relationship. That's how it was done. By the time we got to the 1980s, there's been a few methods developed, kind of perfected, that allowed scientists to look at structures of proteins. And so, of course, the structure of DNA was a big deal, right? 1952, double helix, X-ray diffraction was used to solve the structure. And as you move forward, there were two methods that really drove structural biology. They were called X-ray crystallography and NMR, like you may have done in NMR. So these two methods have really gotten to the point where we can use them now to look at structures of proteins in cells. And so today, 2015, just about every week that a new issue of Nature comes out, Nature is a very top-notch science journal. It's kind of like playing in the major leagues or in the NBA, right? It's like you made it into Nature, you've done something important. So every week in Nature is a new structure of something. Okay. So nowadays we know what the receptors look like very, very well. I'm sure to some degree you've talked about that already in Net 10. How things can fold, how helices can be arranged, how you get spiral structures, or sheets, or loops, or ions coming in and coordinating different loops, all kinds of stuff. So the thing is, proteins have a structural diversity that's unparalleled. Right? DNA, while it's a very exciting molecule because it has all those genes on it, right? It's pretty much stagnant in terms of what it looks like. It's double helix, coils on itself, right? Some maybe coils, more components if you get it. But its structure is pretty much predictable. Right? Not so protein. Right? So proteins have these 20 amino acids that can be arranged in different ways to be different structures. Right? So it's like when I'm downstairs playing with my son with his blocks. Right? It's got all these different colored blocks, and they kind of attach to each other. They make all these kinds of structures. So they'll make some big, unique structure. You say, wow, that's unique. I haven't seen that structure. And then they'll knock it over and arrange the blocks a different way. We'll call it a different structure. Or then they'll arrange it in a more traditional way. Come up with columns and beams and roofs. Right? So in proteins, we have this tremendous structural diversity, which allows us to have special sizes, which allows us to have special types of binding pockets. With sulfhydryl groups towards the inside, or with hydrophilic groups towards the outside. The cell can achieve things it needs to do. Right? Even with this typical receptor that I drew for you on the board, right? We'll call this a cell surface receptor. There's challenges there. right? Because the outside of the cell is watery, the inside of the cell is watery, but the membrane is hydrophobic, right? So if you're being coded by DNA to be a cell surface receptor, there's going to have to be part of your structure that's hydrophobic enough to get you through that membrane. Right? So if you look at those amino acids, they tend to be hydrophobic amino acids. Things like leucine, things like isoleucine, things like valine. They're very enriched in the membrane fraction. If you look on the external portion, right, the extracellular side, now you tend to get ionizable amino acids. Right, there you have a lot of glutamates, for example, or histidines, for example. The same is true on the cytosolic side. And so these different regions of a cell surface protein can be enriched with different amino acids. But the point I'm trying to make here is you've got a lot of structural possibilities here when you talk about proteins. So proteins are pretty complex. If you're to look at all the proteins that are kind of working within a single cell at a single moment, you would call that proteomics. All the proteins that are in a cell at a single moment. Right? It's kind of like the equivalent of thinking about all the people who are on campus right now. What are they doing? How are they functioning? And where are they found inside the university? What is their function? Right? So it's kind of amazing to think, even on this tiny, tiny level of a cell, you might have like 8,000 or 9,000 proteins at a time existing, doing things. Right? It's pretty amazing. So the thing is, we have lots of proteins. So how can we categorize them? Right? So back when I was little, my dad used to buy me baseball cards. He used to collect baseball cards. And I'd have all these baseball cards. And how did I categorize them? I split them into different teams. Right? So I ended up having 26 piles of baseball cards. The only ones I really ever looked at were the Yankee cards. Well, I mean, I looked at the other cards, My dad spent a lot of money. You hardly ever got any Yankee cards in the past. You always got like Rangers cards, right, or Oakland A's cards. Like, were never ever good. Or Jose Canseco, or Mark McFly. So the good thing about getting old is like you got all these cool experiences you can share, right? Isn't it cool to talk to grandpa sometimes? Like he'll tell you stories like, oh my gosh, that's so awesome, right? So you have these different groups of proteins. And the first that we'll talk about are called regulatory proteins. And so if you're a regulatory protein, you're doing something in the cell to regulate its function. Right? You're doing something that regulates function. And so examples of regulatory proteins could be endogenous chemical signals that a cell releases. Things like a neurotransmitter, autocoid, or hormones are all examples of regulatory proteins. Now, that's not to say that all neurotransmitters are proteins. That's not to say that all hormones are proteins. Right? If you think of testosterone, right, that's not a protein. That's a steroid. Right? The structure called a steroid. But if you think of insulin, or thyroid hormone, thyroid hormone is not a protein. But if you think of insulin, it's a big bulky protein. Right? It's released by the pancreas and it travels some distance to the target. And by the way, insulin is a big bulky protein that could never pass through the cell membrane. But it can get up to the cell and it can bind to a receptor on the surface. Very different than a steroid, which is not going to work at a cell surface receptor. It's going to enter the cell through diffusion and do something inside. Right? So we're going to tease that apart in a future lecture. But regulatory proteins regulate. So by definition, a hormone is a signal that's synthesized in one area of the body and travels some distance to a faraway place. Right? So maybe you're made in the pituitary gland and you release into the blood and you travel to some faraway place, like the adrenal gland. Or maybe like the reproductive organs, like the testes. thyroid. You've been released and you traveled somewhere. Had an effect. Distance cycle. An autocoid is a substance released by a cell that gets released by the cell and acts on itself. Auto means self. Right? So it's a signal that acts on itself. So the cell releases it and it binds to itself, regulates itself. Right? So it's very local acting. It doesn't travel a far distance at all. It just travels enough to get out of the cell and bind to a receptor. Okay, so those are all examples of regulatory proteins. And so I'll give you an example of a drug that works like this. One that you often see probably in the pharmacy, injectable insulin. Right, insulin is a regulatory protein. It regulates levels of blood glucose. In fact, it regulates other things as well. It's kind of the ultimate storage signal. Take glucose out of your blood and store it as glycogen in the liver, or store it as glycogen in the muscle when you don't have high levels of glucose in the blood, like right now when you're not eating, or if you go for hours between meals, or if you're fasting, or something like this. Okay, so insulin will be a good example of a regulatory molecule. Enzyme, second type of protein, right? Huge class of protein. Many proteins can fall into the enzyme category. And what makes you an enzyme is if you take a substrate, right, and convert it into some kind of product, right? It's a protein that acts as a catalyst that allows the substrate to convert to a product. And so many enzymes are going to be sites of inhibition for drugs, although not all. Some enzymes may be activated by a drug. But here, let's talk a little bit about inhibition, right? So one easy example to think of is when you have a headache. If you have a headache and you take an aspirin, the aspirin is inhibiting an enzyme called cyclooxygenase, which is an enzyme that makes prostaglandins, which cause swelling and pain in your head region. Right? So by taking aspirin, you prevent prostaglandin synthesis, you inhibit the enzyme. Right? Well, another example our book uses is this enzyme, dihydrofolate reductase. Which essential nutrient do you see in this name? Folate, right? Folic acid, right? So dihydrofolate reductase is going to do something to folates. And folates are important compounds because they get converted into nucleotides called T, thymidines. There's a biosynthetic pathway, several steps, but the end point of the pathway is T formation. So DHFR is required to make Ts. So why would you want to inhibit that? I mean, don't you need Ts in your body to make cells, to regenerate cells, and all that? Perfect. Cancer patients, right? And for exactly that reason. Because their cells have lost control and are growing in an unregulated way. Right? So if you are a tumor cell, you don't care that the patient or the host is going to be affected by the fact that you're growing. Right? So one of the challenges of being the tumor cell is that to make another one and to keep growing and dividing, right, is that you need to also provide a nucleus to all of those cells that has DNA in it. Right? So in order for a cell to divide into two, you have to replicate the DNA. Right? DNA replication. So the process of going from one cell to two cells involves mitosis. Have you learned about mitosis in a previous class? Right? So mitosis is cell division. And it's part of a bigger process called the cell cycle. Right? So the cell, to become cancerous, is going to divide. And as part of that, it needs DNA to divide. Right? So the tumor cell has to replicate its genome. as the tumor cell's replicating its DNA, what kinds of nucleotides does it need? A's, G's, C's, and T's. If you don't give the cell T's, what's going to happen to DNA replication? Is it going to happen well? Is it going to stall? Or is it just going to keep happening without T's? Well, it can't keep happening without T's. It's going to stall. So DNA replication stalls. Right? So by inhibiting T's, you inhibit DNA replication. By inhibiting DNA replication in the cancer patient, is that good or is that bad? Both. It's good and bad. It's good because it's going to slow down the tumor cell, but it's bad because it's going to cause adverse effects in otherwise healthy tissues that do mitotically turn over a lot, like skin, like hair. You ever see the hair fall out of a chemo patient? Right? The inner lining of your gut, right? That turns over a lot, so you can have digestive problems. You're taking this kind of food. So dihydrofolate reductase is the classic example of a target for chemo drugs because it starves the tumor of T's. T's are needed to replicate the DNA. Tumor cells stall. And that's great until what? Until the tumor cell becomes the R word, resistant to the drug, and then it pumps out the drug, and then it starts to go again. Right? So drug resistance is a big problem in chemotherapy. So the drug that our book mentions as an example, and I'll ask you to know drug names as examples of these concepts, is methotrexate. Methotrexate is a DHFR inhibitor. It has utility against cancer. Have any of you seen it in your pharmacy for something other than cancer? So methotrexate is also used to treat rheumatoid arthritis. Right? And so you might say, well, how's that, how is depleting T's effective in rheumatoid arthritis? Well, in rheumatoid arthritis, your joints become attacked, right? Your immune system attacks you, thinking that you are a pathogen. And it starts to eat up the cartilage, it starts to eat at the bone. And so we think, is it a painful process? Yes. People with advanced rheumatoid arthritis, it hurts so much they can't even button their shirt anymore. Like you can't close a print of pain or something. Right? So what's happening there? You're getting inflammation out of control. Inflammation is not necessarily bad. Right? If you cut yourself with a knife and you're bleeding, you need inflammation. It heals the wound and you recover. If you have a scar, inflammation comes at a price. You get healing, but maybe there's a scar now. So inflammation is never free. It's always a price. But its main purpose is to save you against pathogen, injury, and all that. But when inflammation goes too far, you get a problem like inflammatory arthritis. So one of the things that happens inside the joints is that immune cells enter. Right? Immune cells can move and enter sites of inflammation. And there they proliferate. And they keep attacking. So methotrexate slows down the proliferation of immune cells in reducing. All right, the next class we'll talk about are the transport proteins. Now the transport proteins, as the name suggests, move something. Right? They're the 18-wheelers of the truck, of the cell. I live with 18-wheelers every day because I take Route 80 and I take cross Bronx to work. There's 18 wheels every day. They're transporting something. I always look. They're transporting milk. They're transporting coffins. They're transporting bread. Right? They're transporting something. They're moving something from New Jersey to New York. Well, transport proteins are moving something from the outside of the cell into the cell, or sometimes from one place in the cell to another place. And so we need transport proteins. Transport proteins are highly needed to move ions. Because remember, ions can't deal with the hydrophobic portion of the membrane. Right? So they see that, polar head group and they're kind of excited and they're trying to enter the membrane, and once they get past the polar head group, they hit this barrier and they are not able to enter. But yet we need them to enter because of things like the action potential and neurotransmission and excitable contraction of muscle and other things. So ions move across membranes using transport proteins. But not just ions, also biological molecules that are otherwise unable to cross. So take, if you will, glucose. I don't know if you've ever drank soda like Pepsi or Coca-Cola. I actually know somebody who never drank any soda in their whole life. His mom told them when they were small, they would ruin their teeth. So here's this 40-year-old oncologist from Japan who I studied with at Rockefeller. Never had soda in his life. They sing their ass to me. Come on, Koji, you can do it, man. Very disappointing. And he was really into Shinjo. He was a big Mets fan. So I took him to see the Mets once. Shinjo hit a home run. Like this guy who was not very good as a baseball player. He batted like 205 or something. And the one day, I took my Japanese friend, he hit a home run, and it was like awesome. Like texting me back. So the transport works. So glucose. So glucose, right? If you look at Coca-Cola, there's something like 39 grams in a can. Now, if you've ever weighed out 39 grams, it's a lot. Like imagine weighing out 39 grams in the lab on a weight boat and then just eating it all. That's what eating 39 grams. But the point is, you can take that huge, massive amount and dissolve it, and it goes into solution. Right? So it's very hydrophilic. And so that's a problem for the cell, because the cell has a membrane. And so this hydrophilic thing can't enter. But yet, all of our cells use it for energy, right? We all use glucose for energy. That's what all biology is about. We talk about glycolysis, citric acid cycle, oxidative phosphorylation. It was all kind of sugar-based. Of course, there's some anaerobic responsibility. Right? So sugar has to get in. How does it get in? It gets in through a transport protein. Transport protein lets it through. Just can't otherwise enter. And so there are certain tissues in your body where the glucose transporter is present in low levels, but when needed, insulin will put lots of glucose transporters in the membrane and suck in sugar from the blood. Those target tissues are places like muscle, skeletal muscle, liver, fat. Right? So these transport proteins are important. One target for drugs, sodium potassium ATPase pump. Sodium potassium ATPase pump is majorly important in cardiac function. Right? So when the cardiac cell contracts, sodium rushes in, and that's all got to be reset in a few seconds so that it can contract again. Right? So sodium rushes in, potassium leaves, but the cell now is jammed because it's got to reestablish the ion gradient. So it uses this ATPase pump to reestablish the ion gradient. Right? So even this pump serves as a drug target, particularly for a type of drug called digoxin. Right? So even the otherwise seemingly boring transport protein can get in on the action. Even the otherwise seemingly boring structural protein can get in on the action. So structural proteins are exactly what the name suggests. They give the cell shape. And so sometimes these are called cytoskeletal protein. Skeletal referring to the skeleton. So we all have a skeleton, right? We have an axial and an appendicular skeleton that gives us a shape and a form. If we didn't have a skeleton, we'd just be a blob of cells on the floor with a mouth and just kind of be talking from the floor to each other, not being able to move or have shape, right? So skeleton exists in the cell as well, but it's not made of bone, it's made of protein. And it's called a cytoskeleton because cyto means what? Cell. So cell skeletal protein. And there are certain types of cell skeletal proteins that form long strands and give the cell its three-dimensional shape. One of them is tubulin, And tubulin can polymerize into structures called microtubules. Right? So we could call them like bones, if you will. And so tubulin can polymerize into these structures, but the polymerization of tubulin is blocked by a drug like colchicine. So rather than forming these large microtubules, colchicine just causes it to depolymerize. And rather than being nice and spherical, the cells just kind of become blobs that are unable to move. So do you think colchicine is a drug without adverse effect, or do you think it causes some adverse effect? It causes some adverse effect. But it is useful in a disease called gout. Now gout, G-O-U-T, is another one of those inflammatory conditions. It's an inflammatory condition where your immune system attacks a joint, but there's no pathogen there. But what is there is something called uric acid, U-R-I-C, uric acid. Now, uric acid is an endpoint of metabolism in our cells. It's something we normally make, and it ends up being excreted into the urine. Right? Urine is called urine because one of its components is this uric acid. But in certain joints, in certain patients, uric acid crystallizes out. Forms these crystals called uric acid crystals. And the crystals act as a trigger to the immune system. The immune system thinks that it's being invaded. And so the cells enter into the joint and they get punctured by these crystals and they start to explode and release their contents. So more immune cells come and they get punctured, they explode, they release their contents. And all of a sudden, it's massive inflammation in the joint. The joint that usually is affected is the big toe of your foot, and it's usually going to happen, in people who are overweight, who drink moderate to excess amounts of alcohol regularly, and who eat lots of red meat. These are usually the people affected by gout. All right, so colchicine can be used to treat these people because it depolymerizes tubulin in those immune cells that are trying to enter and attack the uric acid crystals. So the immune cells are flat. They're unable to migrate. They're unable to propagate. So even the otherwise boring old structural protein, tubulin, can act as a receptor for drug. So we've seen in the past 5-10 minutes several different classes of proteins and representative members of each class that can act as a drug target. And what we're going to see later in the course are proteins that fit into one of these 4 classes as sites for specific drugs of the ANS. So at this point, I think we'll take a 3-minute break. If you need to go to the bathroom or something. And we'll start up again around 8:40 and go through till about 9:25. Okay, everybody, I'm going to get back to it. So again, we're talking about receptors today. We're talking about how they're involved in the quantitative relationship between drug and response. And on the board a little earlier, I kind of showed you how experimentally we see this relationship. A little bit of drug, a little bit of an effect, a little bit more drug, a little bit more of an effect, eventually saturate the effect. And so even though this approach is what we would call a carefully controlled in vitro system, right? And it's very far removed from the organism, right? If you take a tissue out of the body and put it in the lab on a tissue culture dish, it's out of the body now, right? It's not in the same context it should be normally. If you take cells and grow them in dishes, like that example that I used of neuroblastoma cells, right? You can find a drug that wipes out neuroblastoma cells on a culture dish, but unless that drug wipes out neuroblastoma cells in an animal, it's really useless to you, right, as a new therapeutic agent. So, pharmacology and experimental science kind of grew out of this in vitro approach, cells and tissues, and then moved into an in vivo model. But we need to appreciate that the in vivo model is complex. right? As soon as you put the drug in a patient, the patient's starting to eliminate the drug. As soon as you put the drug in the patient might have a polymorphism in one of its metabolic enzymes and clears the drug. Maybe the drug doesn't work in that patient. There can be things that alter what you think is going to happen based on these in vitro experiments. But the compromise is you got to start somewhere, right? You don't just want to go from never testing the drug to just putting it into grandma, right? So you got to start somewhere. So where pharmacologists start is in these in vitro systems. And so the kinds of things we see over in those in vitro systems could be described as idealistic, right? Under conditions where everything is carefully controlled. And oftentimes these idealistic relationships actually do translate into the in vivo model. But realize sometimes there's a disconnect because there's complex things that can happen in an animal model, that might not be concordant with what you see in vitro. But nevertheless, we start here. And this is where most of the experimental pharmacology began, using tissues, muscle tissue isolated out of an animal, using cells isolated out of an animal, and trying to understand how drugs are going to work. And so again, these words I've said, right, usually as you give a little bit drug, you get a little bit of a response. You increase the amount of drug, you get more of a response, eventually a response. And that curve is a hyperbolic function. And the hyperbolic curve is actually described very closely by this equation. Right? So this equation on the right, where the effect of a drug at a particular concentration is directly proportional to the concentration and inversely proportional to something called EC50. This type of relationship is a hyperbolic curve. And you're going to see the same kind of equation, similar equation in the binding curve. But before I do that, I wanted to reiterate some of the points on the curve. Right? Of course, we have the Emax, the maximum effect. And then there's another point on the curve to consider. This is defined for you down here, but it's called the EC50. The EC50 is a property of a particular drug receptor complex. And what it talks to you about is the concentration of drug needed to achieve 50% of the maximum effect. Right? The concentration of drug needed to achieve 50% of the maximum effect. And it's actually experimentally determined by looking at the graph. Right? So if the maximum effect in this particular example is coming in around 7, right? Then to actually get the EC50, you go down to the concentration that gives you half of that. Right? So half of 7 is about 3.5. And you draw the line over to the curve and drop it down to the x-axis, and that becomes the EC50 concentration. Right? And so by itself, it might not mean much to you yet. But these two parameters are important when we start to talk about things like agonists and antagonists, and when we start to compare drugs to each other. So we're going to build on this as we go. Okay. For now, you can just say EC50 tells you something about potency of a drug, how potent the drug is. Oops. Now, if you were to look at binding curves, right, drug concentration versus binding to receptor, you also see a hyperbolic equation. And so the formula for that equation looks a lot like we just saw for concentration effect curve, where binding of drug to receptor is proportional to the concentration, inversely proportional to something that we call the KD concentration. KD stands for equilibrium dissociation constant. And what the KD is the concentration of drug at which 50% of the receptors, or half max binding, is observed. So you can look at this graph to understand KD. Right, so here's our concentration of drug on the X. Here's our receptor binding on the Y. As we go from low concentration to high, we get a little bit of binding, a little bit more binding. Eventually, we saturate binding. All right, that value is called the Bmax. The concentration that gives me 50% of the Bmax is called the KD concentration. And I just, get it off the graph, just like I did for the EC50. So if my receptor bound fraction is 1, half of that, drop over to the curve, drop down to the x-axis, and there's my Kd concentration. And do you see that the Kd concentration here is pretty much similar to the EC50 concentration in the previous panel? Right? That's usually how it works. Usually you get half max binding, you get half max effect. You get all binding, you get max effect. Except for what type of receptors? Spare receptors. Good. That's what we need to be with. But this is usually what's happening. So the Kd represents the concentration of free drug at which half max binding is observed. Now, do you remember I said EC50 reflects potency of a drug? Kd represents affinity of a receptor for the drug. How tightly the drug binds. So, if a Kd concentration is high, it means it binds with low affinity. I need a lot of drug to bind half the receptors. If Kd is low, then it means that the drug binds very tightly to the receptor. I don't need a lot of drug to bind tightly. Right? Half the receptors are bound at a low drug concentration. So Kd talks to you about how tightly the drug binds, and that's called affinity. Right? So EC50 tells you about potency, Kd tells you about affinity. Now, if you look at this slide, you have two graphs of the same thing. So why do the shapes look different if it's the same thing? Well, the panel on the bottom left is the linear scale. Right? So the x-axis is on a linear scale, the y-axis is on a linear scale. We have a hyperbolic function for concentration versus effect. On the bottom right curve, we have a log scale for the x-axis. Right? So this is a logarithmic scale. The y-axis stays linear, but the x-axis goes to the log scale. So this is called a mathematical transformation. It's the same data, we just transformed it so that the x-axis is log scale. The consequence of that is going from a hyperbola to an S-shaped curve with a linear region in the middle. And in the present day, you might not see the purpose of that. But if you go back in time like 70, 80 years ago, before we had Excel, before we had GraphPad or Sigma Plot or any of these programs, pharmacologists plotted these curves using graph paper by hand. And the problem that they ran into is when they started to compare drugs to each other using hyperbolas, oftentimes the lines overlapped, and it was hard to see any kind of relationships between drugs. So what they did was just a mathematical transformation where they blew up the scale. And they did a log plot on the x-axis. And then they were able to see these straight lines. And one drug would actually shift, the curve would shift compared to another drug, and they could see differences in treatments. So, what's the point of this? Right graph, left graph are the same, but you should pay attention to the x-axes, whether you're looking at Chapter 2 figure in your textbook, or you're looking at something in a future class. What scale am I on? Am I on the log scale, or am I on the linear scale? Because I might be looking at a dose-response curve, but if you're not expecting to see an S-shaped curve with a linear mid-portion, you might not think it's a dose-response curve until you realize it's just a transformation of the same data. Right? Really, it was done for a practical purpose. The problem is that in current pharmacology textbooks, the authors flip back and forth between S-shaped curves and hyperbolas, and it could be confusing, perhaps. So just pay attention to the axes. And it's just a good idea anyway, because some of these curves will be binding curves, so you want to know, am I looking at concentration effect, or am I looking at binding? I skipped that last slide, and I really don't know why, but I'm going to say this, and it applies to that one and this one. This idea of receptor-effector coupling. Receptor is that which the drug binds to. Effector is usually an enzyme in the cell whose effect has changed in some way. It's either been induced or it's been inhibited due to the drug binding. So if we go back to this model here, right, drug binding, effect. How do we go from drug binding to effect? Is the effect right in the drug receptor itself? Are there links on the chain to get to the effect? That which causes the effect is called an effector. Drug can stimulate the effector through the chain. Drug can inhibit the effector through the chain. Is the chain long? Is the chain short? It depends on the receptor we're talking about, the particular receptor. But when we say receptor, effector coupling, we're talking about this. Chain. We're talking about the chain. And so the chain is referred to sometimes as being composed of coupling molecules. Coupling molecules couple the drug binding to the effect. Does that make sense? People get messed up by the word coupling molecule. It's just a molecule that's relaying the signal somehow. Right? You've already learned about one, I'm sure, somewhere in your career, the G protein, right? The G protein coupled receptor. The coupling molecule is the G protein that couples the drug binding the receptor to the effector. So we have agonist, we have effector, and in between is the coupling. And that's what we mean by receptor-effector coupling. And so now we're going to turn our attention to a discussion on full agonists and partial agonists and talk about them in terms of their coupling. So full agonist is believed to be more efficiently coupled to its receptor than, for example, a partial agonist. So what I mean by that, we're going to see in a Now, spare receptors. Our book brings us to spare receptors. These are receptors on a tissue, and they're actually spare in terms of they're not used to achieve the effect, the maximal effect. How can I explain this more clearly? When we sit in a car, we've got 5 tires on the car, but only 4 are on the road, and 1 is a spare tire in the trunk. Right? The spare tire isn't used until it's needed for something. Would you agree? So you actually have 5 tires in your car, but you only use 4 to get your maximum effect. Well, that's like spare receptors. You have lots of receptors on a tissue, but you only need some of them to be stimulated to get your maximum effect. And the rest are considered reserve receptors or spare receptors. Now, what's kind of confusing about them is that they're not actually qualitatively different from a regular receptor. Just like a spare tire might not look different than a regular tire. Right? They can look similar. But spare receptors were demonstrated experimentally in situations where scientists saw maximum response at less than maximum binding. Right? So one way to say that would be if I use the same graph here, maximum binding, right? And in this graph, maximum response. They kind of match each other. But in spare receptors, it might look like this. My Emax can occur at a concentration where I don't have my Emax. Does that make sense? Right, so that was observed sometimes experimentally, led us to this idea that there may be receptors called spare receptors. And so, while it's not the norm, right, spare receptors tend to be the exception to this model I talked about. One very important tissue, myocardium, actually uses spare receptors. And so it's predicted that about 90% of beta receptors in the heart are spare and not needed for the maximal inotropic response. So what does inotropy mean in terms of heart contraction? Inotropy means strength of contraction. So if a drug has a positive inotropy, it means it makes your heart beat harder than it otherwise would. Right? So compared to acetylcholine, does norepinephrine have a positive inotropy or a negative inotropy? Compared to acetylcholine. Right, so vagus nerve is on a lot when you're sleeping. When you're sleeping, is your heart beating hard and fast, or is it kind of... Right? So catecholamines have a positive inotropic effect on the heart. Right? It's just like my wife. She has a positive inotropic effect on my heart. She's like my own personal catecholamine. Right? So positive inotropy means heart beats harder. Another thing we'll talk about later in the course is the rate of contraction. Rate of contraction is chronotropy. Chrono meaning time. Chronotropy. So catecholamines not only cause positive inotropy, they cause positive chronotropy. Your heart beats harder and faster. Your blood pressure goes up. And so yes, my dear wife can at times be a pressor agent as well. But that's okay. You have to have a purpose, right? You have to have a purpose. All right, so the myocardium as a tissue has a lot of spare receptors. And so why? Well, I'm going to say something important. Spare receptors allow strong responses under low agonist concentration. Spare receptors allow strong responses under low agonist concentration. 2. What's my concentration here compared to here? Higher here. Low concentration, spare receptors, similar effect. And so what limits the response isn't really the number of the receptors, but the coupling molecules. You see, in spare receptors, what's limiting is the coupling molecules. So whatever the case may be, I'm at a concentration much lower than the Kd, because if I was at the Kd, half my receptors would be gone. Therefore, the words on the bottom. Spare receptors allow response to be obtained under conditions where the agonist concentration is low. They're kind of like an amplifier. Right? So back in the day when I was a DJ in college, I used to have a radio show on campus, and then I used to do weddings and stuff on the weekends. I would hook up my mixer to an amplifier and crank it up. Right? Be playing some 1990s hip-hop, OPP. You ever heard that song? YMCA. Be putting the wreaths on the old lady. The limbo. Any of you ever done the limbo? The limbo stick and the people going to... All the good memories have come back. But the reason I tell you that is the amplifier, right? So a little signal comes into the amplifier, a big signal comes out. A little bit of drug comes up to the tissue, but a big signal comes out. That's what spare receptors. All right, so the Kd is an important property. It tells you about affinity, right, of a drug receptor. And so if we look at Kd for a moment, we see it's in the denominator. So if the Kd concentration is very low, right, this number is very low, you're going to get a response that's different than if this number is very high. If this number is very high, the whole number becomes low. binding is low. It's not very common. That make sense? Now, the next couple of slides are talking to you about spare receptors, talking to you about this idea of low agonist concentration giving large signal. And I've kind of already said it. I'm going to go through these next 2 or 3 slides, just the words of what I've been saying. Now I want to talk about antagonists and agonists a little bit. So now we're getting to the point where we've acknowledged that the receptor is important for the quantitative relationship. We've acknowledged that drugs bind to these macromolecule receptors and do things. And so what kinds of things can we do? So here's one example. If we have a receptor and there's no agonist around, nothing that can stimulate the receptor is around, and we give an agonist, we get an effect. So an agonist binds to a receptor and stimulates a response. Think insulin. Think type 1 diabetic. They have all the insulin receptors they need in all the right places. They don't have insulin. The pancreas is messed up. Right? So you give them back insulin, and everything is fixed. The receptors are all in the right places, and the signals can go. They just lack the signal. So insulin will be an agonist. Binds to and activates the receptor. An antagonist will bind to the receptor, but it will not activate the receptor. And so it binds to but does not activate the receptor. And so why is an antagonist a drug if it doesn't do anything? Well, that's not true. It does do something. It blocks the signal from happening. Right? So an antagonist is useful when you have an agonist around that would like to bind, but it's blocked. Does that make sense? So you have a headache, and you have all this prostaglandin going on, your blood pressure in your head, and you're throbbing pain, right? That's the agonist. So you take an aspirin, you block it. That's the antagonist. Right? It's working at the receptor. Okay? So an antagonist has relevance if there's an agonist around that's being blocked. Right? An antagonist is kind of like when somebody takes a shot and then you're boxing out to try to get the rebound. You're blocking out the other team. You're trying to get the ball, keep the other team from getting the ball, so that there's no basket. Right? Defensive rebound is an antagonist. And so the effects of antagonists in general result from preventing agonists from binding to and otherwise activating the receptors. We can divide antagonists into 2 classes. Those which are reversible, or also known as competitive, compared to those which are irreversible, also known as non-surmountable. And they produce very different concentration effect curves. I'm going to spend a few minutes. So I'm going to go to the graph, all these words explain what I'm going to say. And so here's a graph, a concentration effect curve, where the white line represents the hyperbolic function we would see for an agonist by itself. And the orange line represents a hyperbolic curve. I tried to draw it with my mouse, but I'm trying to make it a hyperbolic curve. In the presence of a fixed concentration of competitive antagonists. So what you might notice here is that if I increase the agonist concentration from left to right, as I go higher on agonist concentration, I can eventually recover my maximum effect. So in the presence of a competitive antagonist, the curve shifts to the right, but the Emax does not change. I mean, it looks lower, but it's really supposed to touch. So I would just write on the slide, these 2 lines touch each other and are the Emax. I just couldn't get there with the mouse, and I had drawn it a couple times, and it was the closest I could get. So what does it look like when you have a competitive antagonist experimentally? The curve shifts to the right. So if you were to look at the Emax doesn't change, but the EC50 shifts to the right. I need more agonists to get 50% effect, in the presence of the antagonist than I did in its absence. And so this shift of the curve for competitive antagonists results in what we would call a concentration of agonist needed when there's competition going on, or C prime. And we can actually measure the degree of shift as a ratio of C prime to C. C prime meaning the concentration of agonist needed when you have competition around, compared to the concentration of agonist needed when there is no competition around. And that relationship of C prime to C is also known as the Shield equation. And the Shield equation reveals to us that the shift in the curve is going to be inversely proportional to something called Ki value, which stands for the inhibitory dissociation constant. It's the Kd of the inhibitor for the receptor. So if the competitor, if the antagonist, let's say my chair here is the receptor, right? And the inhibitor binds really tight. What do you think you're going to need in terms of knocking it off? You need a lot more agonists to knock it off if it binds really tight, or do you need less agonists to knock it off? Compared to if it were to bind loosely. So if my inhibitor binds loosely, is it easy to knock it off? Right? What if it binds really tight? It's harder to knock it off. What if it binds so tight we can define the Ki as infinity and it's an irreversible binding covalently mediated? Can we knock it off? No. Right? So those are the three kinds of bindings we can see of inhibitor. We can see loose, we can see tight, we can see really tight. The Shield equation is only useful when we talk about competitive antagonists. But it tells us, if we know the Ki of an inhibitor, it tells us how much the curve will shift. So now, in the last 10 or 12 minutes, I'm going to try to conceptually show you what agonists and antagonists look like. So bear with me. In this series of slides, you'll see a bunch of receptors down here in the bottom left. And here I'm at a concentration, I don't exactly know what it is, but it's at a concentration where one of the receptors is bound. So just remember, as these receptors get bound, the drugs are moving in the extracellular fluid, right? They're kind of flowing over the receptor, and the drugs are in that, right? So it's not going to necessarily be every drug molecule binds every receptor. It's going to be a binding in an equilibrium manner. So at a certain concentration, some of the receptors will get bound, and we'll see a little bit of a response. As we increase the concentration, a few more, another receptor might get bound, and we'll see a little bit more of a response. We increase the concentration coming up to those receptors, now another receptor gets stimulated, we'll see a little bit more of a response. As we increase the concentration even more, we'll start to bind up more receptors, and even more of a response. Eventually we bind all the receptors. At this point, what will happen if I add more drug to the receptors? Will I get more of a response? No. So in the next picture, that's what I do. I've added more drug, but the response is max. That's the Emax. And so if you were to look at this curve on the right, I mean, it's my sad excuse for a hyperbola, but it's a hyperbolic function I've drawn with my mouse. So a little bit of a drug, a little bit of a response. By the time I get to saturation of receptors, I get maximum response. And above that, I can't get more of a response. And so this curve just reminds us that the left curve and the right curve are really the same. It's just the scale of the x-axes are different between the two graphs. The left curve is linear hyperbolic function. The right curve is sigmoidal-shaped curve, linear mid-portion, log scale. Now, this slide's out of order, so this slide should be a little bit later. And the next topic I want to show you is antagonist. Remember, antagonist binds to the receptor but does not activate the receptor. So here I'm at a concentration where one of these receptors is bound by antagonists, and what kind of response do I see? Right, that's supposed to be like no response, really. Right, little response, no response. Concentration of antagonist, I bind another receptor, no response. Keep increasing the concentration of antagonist, I bind another receptor. As you may have noticed, antagonists bind to receptors but will not activate them. So I could get a concentration where I bind all the receptors, but there is no response. Now, let's look at reality when it comes to antagonists. Antagonists, since they don't cause response, are only useful at turning down agonist responses, right? So if you ever were lucky enough to go to a big band jazz concert, you might think it's your great-grandma's music. Just go loud. It's like an unbelievable melody. It's like Thanksgiving dinner of music, right? So you see the trombonist. The trombonist has a little thing that they put over the horn called mute, M-U-T-E. So the trombonist is getting all funky, muting the sound, letting the sound go, muting the sound, letting it go, muting it, letting it go, changing it, causing it to sound cool, right? Well, antagonists are like the mutes on a trombone. They reduce the stimulation caused by agonists. So here's a real-life situation where I've got agonists and I've brought in some antagonists. If I had only agonists, I'd have a certain effect. But because I'm competing for binding to the receptor, I've reduced the response. There's a competition going on. Right? It's just like the seat you're sitting in. There were 5 or 6 other students that were seniors in high school that were competing for that seat. And you won the competition. You're in the seat. And they're probably somewhere else or not in pharmacy school. Right? So there's a competition for binding to the receptor that can go on. And so what's going to dictate which of the two binds, I'll tell you about in the next few seconds. So here's the situation. I've got competitive antagonism that I want to illustrate to you. Situation, all my receptors are bound, there's a stimulatory signal going on. Maybe this is pro-inflammatory cytokine, and my tissue is swollen and tender and painful. And so I'm trying to bring in an anti-inflammatory compound. And so anti-inflammatory compound might not do anything in this model. Start to increase it, start to compete. I can eventually compete out all of the agonists. Right, so eventually I've displaced all of the agonists with antagonists. I've turned down the signal. I've muted the horn. But this is a competitive binding. So if I were to now hold the concentration of antagonists constant and start to bring back agonists, look at what happens. Bring back a agonist. Maybe I recovered one of my receptors. Some more, some more, and eventually I've been able to displace all my antagonists. Why could I do it? Because it's a reversible binding. Could I do it if the binding was irreversible? No. Right? So this is what we mean by competitive antagonism.